How's it going, you guys? So today I wanted to talk about the step of oxidative phosphorylation in cellular respiration. Just as a recap, we already went through glycolysis and we already went through the Krebs cycle. Glycolysis made some NADH, an electron carrier, and the Krebs cycle, which occurred in the matrix of the mitochondria, made us some NADH as well as some FADH2. And so all of those electron carriers are going to the inner mitochondrial membrane so that it can carry out the next step, which is oxidative phosphorylation. So all of this is going to occur on the inner mitochondrial membrane. All right, so just wanted that big picture look at it. Now let me move the camera so we can see the membrane a little bit better. So here is my sketch of the electron transport chain, as well as ATP synthase, all part of oxidative phosphorylation, the last step of cellular respiration. Now, if you were to look at this, notice there are a couple of complexes here. There's complex 1, complex 2, complex 3, and complex 4. They all have names, but we won't go into the specifics of those names. Same thing, these are mobile electron carriers. I have Q and site C. This is ubiquinone, and this is cytochrome C. When I say they're mobile electron carriers, they just take electrons from one complex, like 1 or 2, to 3. So again, you don't need to know the names of those mobile electron carriers, but just generally the overall picture of the electron transport chain. So this electron transport chain in oxidative phosphorylation is very similar to the electron transport chain we saw with photosynthesis. So there's something that comes in, drops off electrons, the electrons move through the ETC, and then they're picked up at the end by some other molecule. So that molecule that drops off the electrons in the ETC in cellular respiration, that is NADH. The electron carrier that we talked about that was generated in part in glycolysis, as well as in part during the Krebs cycle. So we saw a lot in the Krebs cycle, some in glycolysis. They come through, they drop off their electrons, so I'll put 2 electrons in here, and now once NADH drops off its electrons, it dissociates into NAD+, as well as H+, a hydrogen ion. Now that these electrons are in here, they can easily start moving through the ETC. They're going to go from generally a high energy area to a low energy area. Because these electrons already are high energy electrons, they don't need a photosystem, they don't need sunlight to excite them. So they will just move their way through the electron transport chain, all the way through complex 1, through mobile electron carrier Q, through complex 3, cytochrome C, another mobile electron carrier, through complex 4. Now, as they do this, through complexes 1, 3, and 4, they are all going to serve as hydrogen ion pumps. And they're going to get their energy from those moving electrons. So as the electrons move, that will provide the energy to pump hydrogen ions. We pump hydrogen ions out of the complexes. So from the mitochondrial matrix into the intermembrane or between membrane space. And now what we're going to see is a concentration gradient. We're going to see a lot of hydrogen ions outside in the intermembrane space, and fewer hydrogen ions in the mitochondrial matrix. So once we generate this proton gradient or hydrogen ion gradient, we can use that to our advantage with this ATP synthase enzyme, which we'll talk about in a second. But first off, we need to discuss those electrons. Those electrons got to complex 4, and now they need something to pick them up. Otherwise, all of the electrons will get backed up, and then the ETC, the electron transport chain, will stop running. So we have oxygen. The oxygen that we breathe, for instance, goes into our lungs, it diffuses into our bloodstream, it then moves to our cells get that oxygen, and we use that oxygen for cellular respiration. Now, this oxygen molecule, if you remember, is small and nonpolar, so it will have no issue getting through any of these membranes, the inner mitochondrial membrane, the outer mitochondrial membrane, even the plasma membrane. It'll just diffuse right through because it's small and nonpolar, and the membrane lets through small nonpolar molecules pretty easily without energy. So oxygen comes in, it will pick up those electrons, and it will be reduced. Remember, if you gain electrons, you are reduced. Here, this was oxidized because it lost electrons. Here, we're gaining electrons, so it's reduced. The oxygen will be reduced to H2O. Now, to be more specific, the oxygen, we technically use 1/2 of an oxygen molecule and 2 hydrogen ions, free hydrogen ions, to generate 1 water molecule. So we really have to go through this process twice in order to fully use up an oxygen molecule. So now that we have the electrons picked up, we can continue allowing electrons to move through. So the purpose of oxygen is to pick up those electrons at the end of the electron transport chain, at the end of the ETC. So this right here, I'll put in, or I will label as the ETC, electron transport chain. Now let's talk about this ATP synthase enzyme. That hydrogen ion gradient that I talked about, we have a lot, or a high concentration of hydrogen ions outside of the, or out in the intermembrane space, and we have a low concentration of hydrogen ions in the mitochondrial matrix. So those hydrogen ions are naturally going to want to diffuse back in. They will go from high concentration to low concentration. And their only way out, or their only way back in, I should say, into the mitochondrial matrix is through ATP synthase. So we use that to our advantage. We use this diffusion of hydrogen ions into the mitochondrial matrix, and the energy that that releases is coupled with the generation of ATP. We take ADP, adenosine diphosphate with only 2 phosphates, plus this third inorganic phosphate, and using the energy generated by ATP synthase, using the concentration gradient, the diffusion of hydrogen ions, we make ATP. And typically for this process, per 1 glucose molecule that we started with, we generate about 28 ATP molecules. Or, oops, about 28 ATP. So if you recall, we made 2 ATP during glycolysis. We made 2 more ATP when we went through the Krebs cycle twice per 1 glycolysis. But that doesn't really matter, or that, I shouldn't say it doesn't matter, but it doesn't generate nearly as much ATP as the electron transport chain or this whole process, oxidative phosphorylation, generates. So really the purpose of the Krebs cycle of glycolysis, the real purpose is to make those electron carriers, that NADH, that FADH2, that will drop off the electrons. Now, you might be asking yourself at this point, what about FADH2? Because I talked about that being an electron carrier, but I haven't mentioned it yet. And I also haven't mentioned this second protein complex here, so complex 2. At complex 2, FADH2 will drop off its electrons, become FAD, and then two hydrogen ions, which will then probably be pumped out into the intermembrane space. But it will drop off its electrons in complex 2, and that will enter the cycle back at that mobile electron carrier Q. Now, one thing you might notice here is that complex 1 is a hydrogen ion pump, but complex 2 isn't. So for FADH2, we technically generate less ATP because we're pumping out fewer hydrogen ions per FADH2 molecule when compared to NADH. So NADH pumps out more hydrogen ions when compared to FADH2 because FADH2 drops it off a little bit later and misses one of those proton pumps. Make sense? So that is pretty much the whole electron transport chain. I want to throw a couple of vocab terms at you just to kind of come full circle. This process right here is called chemiosmosis. And this whole process here is called oxidative phosphorylation. Oxidative phosphorylation. The phosphorylation part happens during chemiosmosis when we add a phosphate group to the ADP to become ATP. And the oxidation part happens during the electron transport chain. So we see it very clearly here and here when we lose electrons. Now, if we were to compare that to the light reactions, we actually call that photophosphorylation because instead of oxidation driving the ETC, it's actually light photo that's driving the ETC. So I believe that is it. Hopefully that didn't make things more confusing. Hopefully it clarified some things. And if you have any questions, just shoot me an email.